It's my favorite subject, marine biology. Didn't have sleep also. Well, it's my great pleasure to introduce our first speaker, Dr. Jan Newton. She is a principal oceanographer with the Applied Physics Lab at the University of Washington, and she's affiliate faculty with the UW School of Oceanography and the School of Marine and Environmental Affairs. She's also the executive director of the Northwest Association of Networked Ocean Observing Systems, NENOOS, and the U.S. IOOS, Regional Association for the Pacific Northwest. Jan is a biological oceanographer. I'm sorry, I'm coffee today. We'll get through this, people. Jan is a biological oceanographer who studies the physical, chemical, and biological dynamics of Puget Sound and coastal Washington, including understanding effects from climate and humans on water properties. Currently, she's been working with colleagues at the UW and NOAA to assess the status of ocean acidification in our ocean waters, which is a huge problem in this area. Jan served on the governor's Blue Ribbon panel on ocean acidification, and she was appointed as co-director of the new Washington Ocean Acidification Center at the UW. And she often is up at Friday Harbor Labs, where it's my also big pleasure to be the librarian up there. So please give a hearty welcome to Jan Newton.
Well, thank you so much for that welcome, and thank you, Maureen. It's been my pleasure to know Maureen over the years when my students need materials, and the Friday Harbor Library is well tended by Maureen. I also just really want to thank you all for what you do. I mean, knowledge is key in our society, and if we lose that, that's a big problem, as you know. And you know, I didn't even plan to say this, but just thinking about what you all do, these are changing times as we go from largely books to so much digital. So thank you for what you do. I know there's lots of challenges. And so it's a real pleasure to be here today. I've got slides and I'm going to talk to you. I was going to show this video at the end, but I think I'm actually going to start out with it because it's a pretty nice video. And, you know, I just can't compete with Cousteau, except when he's silent. So, and I told them, oh, I don't need anything. If this doesn't work, we can show it at the end. But
no.
Yeah, let's just work.
Okay, maybe I should have stuck with my plan. Oops. Sort of lost everything.
So we should have had a disastrous.
Right, I know, it's your theme.
Yeah.
Okay. Well, you know, we're just not going to worry about the video. We'll come back to it, or we will let you look at it on your own. All right, you guys are awesome. Okay, let's start it again.
In the last 200 years, our global oceans have silently absorbed more than 1/4 of the carbon dioxide that people generate. The chemistry of seawater is rapidly changing in a process called ocean acidification. Ocean acidification has the potential to fundamentally change the oceans, its inhabitants, the food webs, and marine life. We need to better understand how acidification affects our oceans so that we can develop tools to slow the rate of change. The U.S. Integrated Ocean Observing System, or IOS, uses real-time data to signal the approach of acidified seawater 1 or 2 days prior to its arrival, providing shellfish farmers with the tools to save their crops.
This is Netarts Bay, Oregon, and we have a shellfish hatchery here.
What happens in the oceans really affects what happens to my business.
The IOS data helps us to manage our hatchery.
We pump in 75 to 100,000 gallons of seawater a day.
We've got 20,000 gallon tanks. As we drain tanks, we fill tanks. We never stop filling tanks.
It looks like dirt,
water, but what you're looking at is 100 million water main right here.
It's a huge responsibility to try to produce shellfish larvae. What you grow not only affects your business, but families out there depend on this larvae to seed their ground. And if you're not producing, you're affecting their livelihood.
So there's a balancing act here. Everything's got to be just right to get these larvae through.
We bottomed out production in 2008.
We would bring the water in, put the larvae in it, and they would die, mysteriously die.
In the back of your mind, you're thinking, maybe it doesn't work anymore. Maybe we lost our mojo.
My wife said, you gotta throw the book out on how you used to grow larvae because it's a new ocean. It's a changing ocean.
I pretty much thought we were gonna lose the business. We never had no survival. That was the difference. We had zero survival.
What drives the pH down is the amount of CO2 that's in the water. On a scientific level, they call it acidification. The larvae hated it, they either slowed down their growth or they died. This is where IO steps in. These guys have the ability to monitor the oceans. This is what helps us run our business, dealing with these changing ocean chemistry.
We used to never treat any water with anything. And now we filter, skim, treat with carbonate, add air and oxygen.
Without the monitoring, we'd be driving blind at night, run off the road immediately in the ditch, we'd be out of business. This is not a localized problem. It goes beyond what we're doing right here. Don't make it a party issue or anything like that. We're talking about the ocean. We're talking about something bigger than all of us put together.
So there, in about 4 minutes, they did what I'm going to talk to you about and give you some more details. So let me see if I can get out of that. Okay, so that gives us some nice, is this on now? Oh yeah, it is, okay, good. That gives us some nice context for some of the logos you see here. So this is IUCE, it's a program that is run through NOAA, but it's actually interagency. It has a federal footprint and then it has regional footprints all throughout the United States, so including the Caribbean, Pacific Islands, Great Lakes, Alaska. In the Pacific Northwest, we have NANUS, that's for Washington and Oregon. And then here's NOAA's Ocean Acidification Program, and they work really well with IUCE to get sensors on buoys and information out. And that's where I'm from. So I have a lot of affiliations, but to me it all sort of makes sense for what I'm trying to do. And I think, you know, we so often stay in these little stovepipes. And when you think about ocean acidification or even other water quality issues, it affects, obviously, the ocean science. It affects having some operational systems to look at. It affects educating the next generation, like we do at Friday Harbor, and it affects societal things like SMBA works on. So now let's back up. So you heard that great video, but you guys are smart. You want to understand what really is this ocean acidification issue. And so I like to use a volcano and a oyster to help tell the story because I think it makes it a bit more real. So what you are looking at here is this beautiful shield volcano. Anybody got to guess which one it is? So somebody said Mauna Loa, but it's actually Mauna Kea because you're standing on Mauna Loa. Yes, exactly. Looking across, trick question, looking across at Mauna Kea. And this is the Mauna Loa station that has been, I'll get it down, guys. There we go. Okay, so it all starts with increasing CO2 in the atmosphere. And so Charles David Keeling started out in the 50s to monitor CO2. And, you know, he's just, you know, I monitor the oceans. He's one of my heroes because he's stuck by his guns to really have this wonderful monitoring system and record what is commonly known as the Keeling curve of CO2. And so you see this is a time series that starts in the 50s and goes, and is maintained through now. This slide is old, sorry. But they're at Mauna Loa. And you see this red line has some squiggles to it. Well, the reason for the squiggles is that's obviously a seasonal signal. And in this Mauna Loa station, even though we think of it as tropical, it is in the northern hemisphere. And there's more photosynthesis that goes on during the summer when it's nice and sunny, and more respiration, that respiration that goes on during the winter. And so that uptake of CO2 is actually the photosynthetic signal in the northern hemisphere atmosphere taking up more CO2 out of the system and then not doing that as much in the winter. So that's actually the breathing of our planet, if you will. And so just remembering photosynthesis is CO2 plus water used for carbohydrates and giving off oxygen. So it takes up CO2. That's what our photosynthetic organisms do for us. So if we just take this last bit here and blow it up. Now this is getting kind of complicated here, but bear with. So this top line here, that red, that's that same data that you were just looking at. That's the atmospheric CO2 trend. They call it PCO2. It's just how the chemists, the P stands for partial pressure, and that's just how the chemists measure and relate the CO2 gas. Now you notice it says of the air or the water, and so the blue line is the CO2, partial pressure of CO2 in the surface seawater, and that was measured at that red dot there called Station Aloha, all right? So it's just north of the Hawaiian Island chain, but pretty darn close to Station Mauna Loa. And so what you do see there is that the red line and the blue line have a very similar trend, increasing trend. So that tells us what the chemists know, which is that when you increase the CO2 concentration in the atmosphere, it's going to diffuse across that air-sea interface and be in some equilibrium. So if you increase one, you're going to increase the other, all right? And that's exactly what you see. The bottom graph here shows pH, and we'll talk more about what pH is, but that's how we measure acidity. And it's the power of the proton, the hydrogen atom. All right, and what you see, while those are increasing in parallel, this is decreasing at about the same slope, but or the same amount once you do the conversions, and it is due to the CO2 signal there. And so let's talk about the ocean's role with the atmospheric CO2, and where does that atmospheric CO2 increase come from? 90% comes from the burning of fossil fuels. So that's either power plants or cars or chainsaws or whatever. So anyways, it's the burning of fossil fuels. 10% of it is from deforestation. Well, why deforestation? Well, because you're cutting down photosynthesizers. What do photosynthesizers do? They take up CO2. So if you have less of them, you have less uptake of CO2. So it's actually a net increase. All right, so once we've got it into the atmosphere, where does it go? About half of it stays in the atmosphere, and that's responsible for the increase that you see in the Keeling curve. And then about equal parts go either into the terrestrial biosphere, the trees and plants, or into the oceans, all right? And then once it's into the oceans, it can be taken up by phytoplankton and a whole bunch of other processes that we'll talk about in just a sec. Go on. All right, so about somewhere between 1/4 to 30% winds up in the ocean. So when we think of CO2, usually people are thinking about climate change. And climate means the temperature, the precipitation, that's climate. We're changing that because of greenhouse gases and all of that. That's climate change. CO2 also is responsible for ocean acidification. They're different processes. They both have CO2 at the root, but they're very different processes. So we're not going to talk about climate change. We're going to focus on ocean acidification. So now that CO2 winds up in the ocean, and I didn't bring the slide that has all the chemistry because it kind of makes people phase over. But anyway, it creates an acidic reaction. CO2 and water actually form carbonic acid, and some people just stop there and they make people think that the ocean's filling up with acid. It's not the case. Carbonic acid dissociates right away. But when it does that, it increases the amount of hydrogen atoms, and pH, it's so crazy. It's the inverse log of the pH. So when you have more hydrogen ions, that means you have a lower number of your pH, all right? We'll talk more about that. And it has a second aspect, is that it decreases carbonate levels, and we'll talk about what that is. All right, so let's first talk about pH. So as I said, it's on a logarithmic scale, and it's good that David Montgomery was here yesterday, although he was talking about the landslide. He's a geologist, and a lot of times they talk about earthquakes, right? And so you all know the Richter scale. Richter scale is also a log scale. And so you know that a little bit of movement on a log scale means a lot. And so this seawater change of 0.1 actually represents a 30% decrease. And we'll talk about that in just a little bit. But anyway, in other words, small changes in pH units mean much larger changes in acidity. And so you'll notice here on this scale, seawater is not acidic. It isn't even neutral. It is definitely basic. And so here comes one of the big communication hurdles that we have when scientists start talking about ocean acidification to the general public. They say, well, I learned in chemistry that seawater is basic, and you guys are talking about acidification. It's not acidic, and it's not even going to an acidic number. But acidification, the way that the pH scale was made by the chemists long ago, is that it wasn't set up to measure basis. It was set up to measure acidity. And if you're going in this direction, it's called acidification. So the analogy we use is you're here in Seattle, and we get one of those cold snaps, and you're down at, at 28 degrees Fahrenheit, and there's ice everywhere, right? And so then the next day, it warms to 34. Is 34 warm? Heck no. Is seawater acidic? No, but it warmed and it acidified, okay? So that's just how we use the language and the concepts, but it loses the public, or a sector of the public, a lot, and that's a problem with communicating this, so I wanted to tell you that pitfall. All right, so ocean acidification. CO2 added to seawater changes the water chemistry, reducing the pH and the carbonate levels in the ocean. And here is basically showing with data, and of course this is schematized, but it's based on references here, showing what we've seen through to the year 2000. And so that 0.1 change in pH is a 30% increase in the acidity and a 16% drop in carbonate. And we'll talk about significance of carbonate coming up real soon. But the scary part of this whole thing is you can see by those slopes that we're really turning a corner, and this is where we're headed. And that's the real sobering part, is that if the emission scenarios stay as business as usual, these are the predictions for the year 2100. And so 150 to 200% increase in the acidity and a 50% drop in the carbonate. So those are some pretty serious numbers and effects. And as you heard from the shellfish grower in Oregon, we're already seeing effects now. All right, so let's hear more about the oyster's perspective now that you've understood the CO2 story. So this is a paper, an article from the Seattle Times that came out about 2006, I think it was published. I used to have the date on here. Anyway, noting that oysters are not settling like they used to. Oysters are grown in two different ways commonly. One is what they call natural set. The larvae are just out there, they're swimming around, and they settle, and they oftentimes settle on the shells of the other oysters that are either still there or just open shells. So the oyster isn't there anymore, but the shells are, and that's called natural set. The other thing is what Mark Weigert and his wife Sue Cudd were doing, where they're actually incubating larvae like you would grow seeds, right? So like a seed bank, and so that's a shellfish hatchery. So then they produce the larvae, and then that larvae gets sent out to different shellfish growers who put it in their tidelands, and it seeds the tide flats. And so those are the two different methods. And so what was really interesting is hatchery growers were having issues, but also the natural set was going away. And it was going away multiple years. And so people were really trying to figure out, is this a virus? What is it? Much like we're trying to understand the starfish wastings. And there's no understanding that that's linked to OA. So I don't want to give you a confusion there. In that case, they're still researching what it is, but it's unlikely that it's OA. In this case, people started to wonder. And the reason why they wondered is, okay, so now we're going to finally focus on carbonate. So we see that when we have ocean acidification, carbonate levels are reduced. So how does that figure into the story? Well, carbonate is a building block for calcium carbonate. What is calcium carbonate? Oyster shells, right? Okay, so that's that white chalky stuff. It's in all sorts of marine organism shells. And so if you don't have as much carbonate, you're not going to form as much calcium carbonate. And so the chemists have, or chemistry has devised equations to figure out the saturation state of calcium carbonate. And it's denoted by that little Greek symbol omega, all right? So omega, if it's at 1, that's the equilibrium concentration. If it is below 1, it's really difficult to build shells because the chemistry equilibrium is for the calcium carbonate to be in the dissolved form and not form that solid. If it's above 1, then it's relatively easy to build shells because it's favored, that precipitation is favored. Okay, so that's the whole thing. And so, you know, part of what I wanted to communicate to you is what ocean acidification is and what we see in our Pacific Northwest waters, but also what are some of the pitfalls in explaining it. So pitfall number 1 is what I told you about the pH business. Pitfall number 2 is how many people, you know, or aragonite saturation state was less than 1, you know? It's sort of like, sure. Oh, you're just used to omega. Right. Okay, so it isn't, these aren't words that people are used to hearing, but when you say how easy is it to build shells and are you above a threshold or not, people can grasp that. But you have to understand the underlying science too so that you can explain it well. All right, and then it gets more complicated because of course things aren't linear. The calcium carbonate system in seawater is really, really complicated and nothing's linear. And so pH is not linearly related to saturation state of aragonite, not perfectly. And so, so it's hard to go between the two. But when we're at the threshold of 1, pH is around 7.7, which is a lot lower than 8.1. But you'll see as we go into coastal systems, it's much more likely that you'll have some of those very low pHs, both naturally and then for other reasons. Okay, so does this omega really matter to the larvae? So here we have omega in initial water, and you see it even starts at 1 and then goes above. So shell formation, not very possible or probable when you go below 1, but even above 1, if we look at these experimental results, production is lower and growth in feeding stage is slower. All right, well maybe you're not used to thinking about those production and growth in feeding stage. But look at this. This picture tells 1,000 words. These are oyster larvae. When the larvae settle, the first thing they do is they lay down aragonite and they make this little thing that's called a D-hinge for obvious reasons. And so this one, these two, day 1, day 4, was grown in conditions of 1.64 omega, and this one was grown in 0.47. So you see, it actually can make a shell, but it's not very good, it's deformed, and because of the energy expenditure, it's probably not going to be so likely to develop all the way. So it's all about energy budget. Having a lower aragonite saturation concentration just means it's harder to do it. Conditions are determined against you. All right, so let's just review. So atmospheric CO2 has increased from human activity, the combustion of fossil fuels primarily. We know that oceans absorb CO2. We know that increased CO2 causes ocean acidification, and this changes the water chemistry, reducing pH and carbonate levels. And we know that OA, that process, that reduction in carbonate levels and pH, reduces growth and survivorship in some organisms. Okay, particularly calcifiers. But this is the real scary part, if that isn't scary enough. In systems, it's all about the rate, right? You know, if you're losing some of your budget every year, it's okay. But if that rate increases, it starts to be really disastrous. Well, if you look at the carbon dioxide concentrations over history, you see that they were rising, and then you start to get into this exponential increase. And I love keeping this slide in because I used to actually use it when we were in the year 2000 or just beyond that. Now we're in 2014. I updated it to 2012. It was 391, which is well off this graph. Now that's wrong. We're at about 400, okay? So this is happening in our lifetime. It's very real, and it's very sobering because it's showing us that this rate has a rapid rate and no parallel in millions of years. And that's, to me, as a biological oceanographer, I know that organisms adapt. They mutate, they adapt, and we've got organisms with short lifetimes, so some of that can happen rapidly, and there's always niches, and you'll have winners and losers and that sort of thing. But when you crank that rate up faster and faster, that's when you start to really stress systems. So that's the part that I think should garner our concern these days. All right, so many biological processes are sensitive to changes with OA. These are results that are still being researched. There's a lot of activity now in looking at different organisms and looking at the OA stress and seeing how they react. So we talked about oysters. This little critter is called a pteropod. Once again, an easy concept, pteropod. Jeez. So anyways, it's got this aragonite shell that it builds, and so this is a microscopic organism, like if you're familiar with copepods maybe, but anyways, it's one of the plankton of the sea, one of the many plankton of the sea, and it's otherwise known as a sea butterfly because those two appendages that you see, they kind of flap like wings, and they're very beautiful. Anyway, they're plentiful in plankton, and so there's been some research on how OA affects pteropods. And so this was done in the lab. And so all of these things here are kind of lab results and extrapolations to nature. But just recently, in 2014, earlier this year, Nina Bednarsek, who's a postdoc over at NOAA PMEL with Richard Feely, who's one of the preeminent leaders of the field of ocean acidification and understanding it in our world's oceans. She did research on shell dissolution. And so this is a photomicrograph now of this shell, and you can see how it's pockmarked and starting to sort of dissolve away. And she looked at it in what's known as the California current system, and that is a current that as the waters come from, like, say, Japan over to North America, then the southward current that goes from pretty much Canada to Mexico is called the California current. So this is the current that's off of our Washington coast. She found dissolution especially as you get to the coastline. All right, and so we're hoping to use this as an indicator of ocean health. Well, there are some organisms that may benefit from this increased CO2 availability, and that would be the photosynthesizers. So things like kelp, seagrasses, ulva, seaweed could, they need CO2 as their food, if you will, to make photosynthesis and happen and to make their biomass expand. So for their growth, they need CO2. And so a high CO2 world might be good for some forms of photosynthetic organisms. And moreover, we may be able to exploit that or utilize that to mitigate local effects. And so that's where some research is going into it. We had these big rafts of algae. Could we offset the pH in a localized area? Now, the jury's out because the experiments haven't been done, the field observations haven't been done to see if we can do that on local scales. On a worldwide scale, though, very unlikely, because when you think about the volume of the ocean and the surface area of the ocean, it's way overwhelming. All right, so just kind of recapping here what we've talked about. We talked about this CO2, and we talked about the rate, and we talked about that it's faster than what's been in recent history. Okay, so now you know the underpinnings of ocean acidification, what it is scientifically. So I like to say, and I think it's really important view, is it's a global condition, but it has very local effects. And let me show you these two slides next. So I showed you the time series from Hawaii. This has PCO2, pH, and carbonate. Hawaii is a time series in that golden color, but there's a data series from Bermuda that's green and Canary Islands that's blue, and you see in the world oceans, they're all doing the same thing. So this isn't just something spunky happening off Hawaii. This is that our global oceans, this is a global condition. But here is a very local effect. And many of you are not from here, so you wouldn't recognize, but there's two people in this picture. The person in pink is our former governor of Washington State, Christine Gregoire. And the man wearing jeans is Bill Dewey with Taylor Shellfish, a very active shellfish company in our Puget Sound and Washington area. They have a hatchery similar to the one that Mark Weider and Sue Cudd run down in Oregon. And the shellfish growers industry really are seeing the effects of ocean acidification currently. You heard from what Mark said in that video, and Sue said, we never had no settlement. That's what was really different. And so now they're treating their water, trying to raise the pH and increase the carbonate levels, and that's making a difference. So they knew something's going on. So their industry was active in talking to decision makers, including Governor Gregoire, including Congress folks, and so because it's really impacting us locally. Ocean acidification can have local effects all over the world. Coral reefs in the tropics, certainly shellfish growers in Africa, in Asia, in Europe, they're all going to see it, but it will be influenced by the localized effects there, okay? And what's really important is we need to get a handle on those local effects. And the oceanographers have been measuring ocean acidification in the global ocean, in these big open ocean settings, and not as much in these localized areas. And that's the tide that we're turning now, is to try to get better information here locally. And so Governor Gregoire assembled a blue ribbon panel in 2012, I guess it was, that produced two reports. One is this panel, which was composed of about 40 people from decision makers, scientists, NGOs, industry reps, tribes, other government folks, to create a document that made recommendations. Let's go from knowledge to action. And then this is the scientific summary that's Appendix 7 to this. Both of these are available online. The URL's kind of long, but if you just Google ocean acidification blue ribbon panel, you'll get there. And if you have any troubles, don't hesitate to contact me. I'm pretty easy to find. I mentioned at Feely, Terry Klinger is also at the University of Washington, and as Maureen noted, we are now leading the Washington OA Center, and then May Chadsky with Seed Grant. So there's a lot of really good material on that scientific summary in Washington waters. There's a similar panel now that's been established for the West Coast, California, Oregon, Washington, BC, and other states, Massachusetts, Maine, Maryland, a lot of the M's, are doing similar sorts of legislation. So what did we find? Well, OA effects are appearing in Washington sooner than anticipate, but it's for some very local reasons. Some of it is this process called upwelling, and I'll show you what that is in a minute if you're not familiar, and the fact that we have a very productive situation already, and I'll explain how that affects this whole phenomenon. So here is how upwelling works. So when the winds flow from the north to south, so equatorward winds, because the Earth is rotating and the Coriolis effect, the water is displaced off the coast and deep water comes up to replace that water that's been displaced off the coast. So this happens the world over, upwelling. But it happens very strongly off of the west coast of continents. And so if you look now at some data, and give me a moment to explain what this is. These are data from the west coast of the US and data from the east coast of the US. This gray thing, so what you're looking at is depth on this axis and distance from shore on this axis. And the gray thing is the seabed. And so right away, you can see that the continental shelves on the west coast are very much narrower than on the east coast. If you look at the bathymetry map of the US, you'll see that. But the other thing is that the equatorward winds on the west coast are going to cause this upwelling, whereas that won't happen on the east coast because the water's going in a different direction. So now the data are salinity, oxygen, and our friend omega aragonite. And you can see in every case during the upwelling season, which is largely summer now, this deep water is coming and bending upwards. And so that is basically because of the displacement of the old surface water. It used to be level like that. That got pulled off of shore, and this comes up. And so so high salinity, low oxygen, and low omega aragonite. So interestingly enough, the signal from the atmospheric record from OA starts at some values, or depths right around 50 to 100 meters, and then we're combining it with this already naturally low CO2. So now you've got in these waters the anthropogenic plus the natural signal. So we're kind of stacking the deck, all right? The other thing is that we talked about photosynthesis, and it takes up CO2. The other process is respiration. That's what we're doing right here, breathing in oxygen, expelling out CO2 and water, and metabolizing. Respiration releases CO2. Now, you'd think that all the phytoplankton in the world growing with all this nice CO2 at the surface would be great because it's going to suck the CO2 out of the surface seawater. True, except what happens to that biomass? It sinks, it gets consumed, and then those organisms sink, or they poop, and that sinks. And, and so you've got all this organic matter down here, and bacteria love it. What do bacteria do? They consume it, they respire, they produce a lot of CO2. Our waters are naturally productive, so we naturally have high CO2 waters. And as you get inland, this deep water is more easily injected into the surface because it's just shallower, right? All right. I'll come back to there's a funny joke. I'm so glad to hear that you guys are going cruising tonight. Anyways, but it's a good joke about water depth. So then you have to ask, are local stressors contributing? So we know that we have runoff, we know that we have fertilizers that go in, we know that we have gas emissions and all of that. Is that making a difference to our local systems? Jury's still out on this. It could, but no one has done the calculations. They're very difficult. They rely on having a lot of good data, operational models, and we're in the process of starting this investigation locally here in Washington. And so that's an important question, because if we control some of these things, will we buy ourselves some time for this problem? Here are some data from, okay, so what are we looking at? So this is CO2 concentration, and it's now called XCO2, not PCO2, for sublime chemical reasons I'm not going to go into, but it's CO2 concentration. The green is from a buoy, and this is atmospheric. The green is from a buoy off of the Washington coast near Cape Elizabeth, which is in the coastal ocean. You don't see land out there. And you see this nice seasonal pattern that we were talking about, the uptake during the summer, and then the relaxation of that during the winter. The blue is a sensor that NOAA PMEL put on the top of the Space Needle. I know you know where that is because it's on Eli's shirt, and you got them all too. So you see there's two things that we're learning from this. We can see that increase from the global signal off of our coast. But the other thing is, look at the Space Needle data, and you see these really high spikes. Well, what is that? That's automobiles, that's industries, that's all of the human effects. These are not that far away mileage-wise, but obviously sampling different air situations. Now, atmospheric CO2 gets mixed really quickly. Is this having an effect on our local waters? Don't know. It could. Calculations are still in the mix. And Dick Feely and his group, Simone Allen, and others are really leading that effort. All right, so this is really some of the key questions that the scientists need to be able to answer as we go by. What determines these local nearshore levels? How can we get them measured more easily? Because it's really difficult right now. Could we have management processes that reduce local OA? How fast and how far can species acclimate and adapt? Are there crucial tipping points? Which ecosystems are more or less resilient? So these are all areas of active research that we need to get a better handle on. So, but there's a lot of challenges. As you go inland, because you have strong photosynthesis, you have upwelling, you have mixing and all of this stuff, the inland water CO2 carbon chemistry has a lot of variability. As you know, looking for a signal when high variability is happening, this makes it more challenging. Source attribution is difficult, but you can do it, and I'll show you an example of that. And then we really don't understand the biological aspects. People have done a lot of research on oysters. We need to go beyond that. So here is some what we'll call source attribution off of the coast. So you can see it's broken to coastline region. And so if we look at the acidification signal that we see, how much is due to upwelling, respiration versus the global CO2 emissions. And so you can see upwelling right now, definitely the bulk. Respiration, sizable. But global CO2 emissions is definitely there. And the problem with this column is this one's going to change. These two will likely not in terms of their capacity, but the percentages, of course, will change because as this one increases, these ones will go down. So that's where we are right now. And as we look inland, we have all these processes going on. And as we sat down with the Blue Ribbon Panel report, we thought about all of Washington's waters and how they're not really the same. You'll look out at that beautiful blue Puget Sound, and it's just gorgeous. But if you were out on, let's say, Willapa Bay or in a different place, it might look the same. You're just looking at that beautiful blue surface. Willapa Bay's only about 20 to 10 meters deep. Puget Sound's 200 to 100 meters deep, and here comes the joke. So, so I was part of a conference that was in Seattle and went out on this boat. I don't know which one you're going out on, but anyway, there were some folks from Florida, and this one woman says, "Oh my goodness, I've never been out on these waters. You know, how deep is it here?" And I said, "Oh, about 200 meters." She goes, "Oh my goodness." I said, "Well, you know, you can drown in 10 meters. I'm not sure it helped. So, I know your boat is very safe. But I'm really happy that you guys are going out on the Puget Sound tonight. It's going to be great. And it's so great to hear you talk about a cruise, because when I go on a cruise, I'm filtering water and working 12-hour days. You guys are going to have a really nice time. And we call it the same. I still call it a cruise. That's how I'm going to be at sea all next week, all throughout Puget Sound. So it's cool. Anyways, all these distinctive influencing factors, I'm not going to go into, this isn't a local talk, but the biology exists within those environments and faces different pressures, and we've got to get a better handle on that. So what do we know in the Pacific Northwest? We know some of the things that we've been talking about more generally, but we know that that's global and affects our Washington waters. Here's the real kicker is, so now we've been talking about all the anthropogenic cause of change in omega or pH or CO2. There's also all these natural factors, the upwelling, the respiration. And so these data are kind of complicated, but they're different years. This is saturation state on this scale. It's all put on an annual basis, the different years. And you can see the upwellings times of years, right? It's right around summertime. And this is surface seawater. And so when the upwelling means it's bringing deep waters that have the lower values to the surface. So that's why it's so spiky. Upwelling happens and it shuts off, etc. But this line right here is the 1 omega line. And this paper by Harris, who's a graduate student who was working with Bert Hales at OSU, another really strong leader in this field, she calculated that the percent of time that we cross that threshold now is 33%. Pre-anthropogenic CO2 emissions, it was 11%. So here's the saying, it makes a bad day worse. So imagine having a bad day 33% of the time instead of 11% of the time. So even though the anthropogenic signal is much smaller than the natural variation, it's additive. It's still making us cross that threshold. And a lot of people get bogged down because they say, oh, it's a small signal compared to the natural variability. True, but it's additive. It's not comparative. All right. Small changes can cause large responses among living organisms, and we are seeing it locally. Enough said. So, on the heels of this report being published, our legislature here in Washington State did 2 things. They established a resource advisory council, Marine Resource Advisory Council, of similar kinds of leaders that were on the Blue Ribbon Panel. Martha Konsgaard is the chair of that. They meet regularly and are talking about where do we go from here? What recommendations should we be acting on in the next biennium? Our state works in 2-year biennium. They established this thing called the Washington OA Center at UW, and our dean, Lisa Gromlett at the College of the Environment, appointed Terry Klinger and myself to co-direct that. And the legislature gave us money to implement 5 action from those recommendations. And so here are those 5 actions. So continue working with the shellfish growers to get the water quality monitoring in their hatcheries. And then expand and sustain OA monitoring, ocean acidification OA, monitoring throughout Washington's waters. And so a lot of partners involved in that. Conduct some studies on Washington species and ecosystems. And we're doing that now with Dungeness crab, with gooey duck, and with copepods and krill from Puget Sound. And then to use our best models to make short-term forecasts of corrosive conditions. So much like you get a weather forecast, we know that being able to predict things like upwelling, or when we're going to have a lot of productivity, or when we're going to have oceanic intrusions, we should be able to forecast that. So it's a very exciting development going on with some modelers. Parker McCready here at UW is leading that effort with others. And then also to give advice to the growers on different methods or designs that could help them grow larvae more effectively. So those are the 5 that we're implementing. As I mentioned, of course, we're excited about being at the cutting edge, but we are seeing a lot of other states in the nation do similar things. And also, we're not alone. We're in our federal context. And so we're joined by NOAA and EPA on these things. And so that brings us back to the US IUCE. And Here, I'm happy to say that we've been working, the University of Washington and NANUS have been working with NOAA for many years. We were deploying this buoy in 2010, and it's off of La Push. And if you go online right now to NANUS, which is www.nanus.org, and look for a buoy called Chaba, which is written right here. It's C-H-A-B-A. We deployed this off of La Push, where the Quileute tribe has their local and accustomed land, and we worked with them, and one of their tribal members named it Chaba, which means whale tail. And she said, like when a whale dives, you see the tail, but there's so much below that. Well, when I look out and I see this yellow buoy, I know there's so much below that. So that's Chaba. In case you didn't know Quileute so much, but anyways. So you can click on either, find Chaba, La Push. It's just kind of like right off the Olympic Peninsula. And you'll see the real-time data from this buoy being transmitted. And so not only do we scientists look at it, the shellfish growers look at it. And Mark, the guy who was in the video, who's so great, he's always giving us these great quotes. It's like putting headlights on a car. So that's an exciting development in terms of being able to get information for people to make better decisions. And so here's actually, I showed you this, but anyways, Chaba's out over here. This happens to be a data display from Taylor Shellfish, so the shellfish hatchery that Bill Dewey is involved in over here in Daybog Bay. And what's really exciting is when we post that data, then it shows up over here on the USIUS site. So it's all networked. And then USIUS is a partner in the effort to build a global ocean acidification observing network. And so this is something I've been putting a lot of energy and excitement into, is working internationally to get this ocean acidification information on a global basis. Because, let's see, I have it, yes. Always global condition with local effects. We need to get local through global scale observations in order to get either correct. If we don't know the global context, we'll never understand our local situation. But conversely, if we don't understand mosaics of these local effects, how can we describe our global condition? We can't just leave that to ocean values because they're different. So we need to get both of them. And so we really need to be coordinated, and that's where the Washington OA Center is one of those coordination points for us locally to coordinate science efforts, regional focus, connect decision-relevant science with managers and policymakers, promote outreach and education, and engage broadly with others. We do that on the local scale, we do that through IUS, and we do that through GoOn, and we're starting to get that network that can start to really have some impact on people's awareness and knowledge in order to make sound decisions. These are the folks that we are partnering with just in Washington State to get the monitoring done. And just, I think there's 16 some odd here. And it's exciting to really be working with these people collectively. Here's some of the tools we use. We're just on this big ship. Yours will be much smaller. From San Francisco to Seattle. It was sponsored by the National Science Foundation to look at harmful algal blooms, but the scientists invited us on board to do the OA measurements. The next panel down shows the intertidal. We're working with the Olympic National Park and the Padilla Bay Research Reserve folks to get sensors in those nearshore conditions. We're also deploying these big buoys in Puget Sound, so similar to Chaba. So now here you see our team working to instrument these buoys with the CO2 sensors with NOAA QMEO. So anyways, ending with the last slide here, I just want to say that it's really an honor of my lifetime to be involved in this Washington OA Center here at UW, working across many scales with many partners to to try to make a difference in the availability of data, information, and coordination. And so that's the website. It's not a stellar website, I'll warn you, because we've been busy with other things. But if you want to Google it, you can find links to the Blue Ribbon panel, as well as those 6 things we know about OA in the Pacific Northwest and a lot of other resources. So I am really looking forward to your questions, so I'll end it there. Thank you. Yes?
Yeah, I just had a question about gathering data like out in the open ocean and further down in the water column. It seemed like a lot of the data you're collecting is in the shallow or coastal surface waters. What do we know about what's going on out in the open ocean?
Yes, so the question is, what do we know about what's going on in the open ocean aside from the surface? Because all the data I showed was surface. There, let me go back to, it's a really good question, and not enough. See, each of these lines, let's see, the blue lines, ignore the green lines. The green lines are the volunteer observing system and ship of opportunity cruises. That's just surface measurements. But these blue lines that you see, those are ship-based time series hydrographic cruises, and they do full water column profiles. Now the only problem is they only do it about once every 10 years. It's expensive. Yeah, so that's what we know about pH and carbonate at depth, is really reliant on those sections, decadal sections. We are starting to get buoys that have sensors at depth. In fact, this September, we're going to be deploying one on Cheba and also in Puget Sound. And part of it is just, you know, the electronics of the sensor. So there's, you know, there's pH sensors. You can go buy a pH sensor for, you know, a few $100s, but it isn't of the precision or accuracy needed in order to make OA relevant things. So the ones that are really good cost about $20,000, and they're better at the surface, but they're starting to make some that are rated now to 70 to 90 meters, and so we're going to be field testing them. And then there's an organization called the X Prize, and they offer prizes for development and knowledge, and so they're doing a technology X Prize for a better pH sensor. So we're really excited about this. It's going to be tested here at Seattle Aquarium. Dick Feely's involved in that. There's going to have other test sites in Hawaii and a few other places. So I'm very hopeful that if I was answering your question 10 years from now, that we would have more information. But we're very limited right now. Yes, in the back.
Okay, can you hear me okay?
Yes.
Okay, great. My question is centered a little bit more toward the Texas Gulf Coast, where I've lived for a long time. I worked with a student there who was studying dead lakes that had been killed due to the algae bloom, and I think that's called eutrophication.
It is.
Okay. Those kinds of situations in our freshwater land-based water systems that eventually would join the larger water picture, are those source attributions, or are they looked at in a way to talk about this kind of an issue?
Yes, so the question is on lakes that have been eutrophied. That means that so many nutrients have gone into them that the photosynthesis is just cranked, and then finally all of that stuff has died and it just becomes anoxic, and nothing really grows in it because it's an anoxic environment. Anaerobic bacteria love it, but there's no oxygen there anymore. And it can happen in small systems like lakes because there's no really good way to replenish it. So the question is that making an impact? Can that be a source of impact on OA conditions? And the answer is to local bays that, for instance, if that water ran into, it could have a localized effect. The big thing, though, with our land-based systems is, you know, even big lakes are really tiny. I mean, okay, so look at the Great Lakes. Look at them on that map. Do you see them, right? You know, but on the other hand, if you had a river that was coming into a bay that was as big as this room, then of course, yeah, you're going to have some localized effects until the bay that's as big as this room flushes and exchanges and mixing with the ocean that it's connected to. And that timing is different depending on what kind of a bay it is. Does it have a sill that inhibits mixing? Is it, you know, are you in an area where there's really strong tides? And so that's what I mean. It's like these local effects get really important. So certainly something to look at a local basis. Yeah, over here, and then Eli.
Do the polar ice caps and the melting glaciers qualify as local or global? The scale of those?
Yeah. So the question is about the melting ice caps, and is that a local scale or a global scale? That's definitely a global scale phenomenon. And does it have a linkage to ocean acidification is a harder question to answer. But when you have more fresh water in the surface oceans, you're going to have the possibility that it's harder to mix the water, and that people are still trying to decide whether there will be enhanced productivity or reduced. Most people think it will be reduced because what do phytoplankton need? They need nutrients, they need sunlight, and then they need a certain amount of stability, right? Because if you're in a water column that just gets mixed and you're taken out of the sunlight, then you can't grow. But if you have a certain amount of stability, the oceanographers call it stratification, then you're happy, as long as you've got enough nutrients. But see, that's the thing, the stratification can cause the nutrient supply, which usually comes from depth, to be cut off. So likely we're going to see less productive systems and systems that don't mix quite as much. Now, how will that have an impact on OA? It will tend to keep high CO2 waters maybe more restricted to depth, but that means that when they upwell, they're going to be even more potent. But everything comes down to a scaling, and I don't know if those effects are going to be significant or not. Certainly, what you said is a global scale effect, but how it impacts OA or not, I don't really know yet. Yeah, Eli.
I was wondering if you could talk a little bit more about some of the communication parts of your work. I was interested to hear you talk about, you know, the challenges in communicating these efforts, and I know that you're in some fairly, you know, public roles, and sort of communicating with the public, particularly in light of the challenges of communicating with folks in the public who have a perhaps particularly closed mindset or a very ideologically based mindset. Those who've been following the Twitter feed may have seen that we've been joined by someone not at the conference who is trying to argue that ocean acidification is a fraud perpetuated by the government. So how do you respond to those kind of voices?
Yeah, so, right.
And do you get that all the time?
Sadly, the answer is yes. So how do we deal with that? I think the thing that I really try to do is stress what's based on chemical laws. I mean, you can't argue equilibration of saturation states. You can't argue dissolution of gas across the air-sea surface. Those things happen, and usually the doubters are falling into a couple of the pitfalls that I mentioned. One being that they don't understand that what we're saying about acidification, meaning that it's going in, and some people talk about, should we change the name? But then that almost sounds like you're kind of trying to evade and, we'll call it something else. It's like, no, this is really what it is. And so we try to use the warming analogy to get over that. The other thing is, a lot of people are really harping on, well, the anthropogenic signal is so much less than the natural variability, and really stressing that it's additive. And I think that very simple statement, and I don't actually know whether Burke said it or I said it or somebody else said it, but makes a bad day worse, is that's helping people to get it. Because we all know that happens in our lives, and so just thinking about changing that. And so, you know, one audience at a time is kind of what I'm involved in. But then we try, there are these FAQs, frequently asked questions, that we just produced, and they're, I don't know if they're on our OA Center website yet. I know they're on the NANU site. And so working to try to get some of that communication material out. The problem is it's just reading a document. Most people are just going to say, you know, huh. So I don't know. Well, so, okay, so here's, I think if the shellfish industry wasn't engaged in this, we'd be nowhere. I wouldn't be standing here in this room. There would not be a Washington OA Center. The governors, both Gregoire and Inslee, our current governor, would not have put the support and focus on it if they didn't see that it's affecting economies. And you know, it's so funny when in that video, Mark Weigert says, don't make it a party issue. I thought he meant like at a cocktail party. I know about ocean acidification. He thinks it's a political party issue. So you can tell what I spend my time thinking about. So what I have heard from going back to DC and visiting our legislature in terms of, you know, giving them information, we're starting to get more traction across both sides of the aisle. And Representative Herrera Butler co-sponsored with Representative Kilmer a bill to try to get better technology and research on ocean acidification, again, through a prize mechanism. But so there's an R and a D working together. And I think because it's hitting us in the industry, and I think there was just a climate report that was published by people from industry talking about how climate change and ocean acidification will affect industries. I think that's starting to get some recognition out there, because people don't want to, your gentleman doesn't, or whoever that person was, doesn't want to listen to a pointy-headed academic like me. They want to maybe believe some guy on Wall Street saying, this really is having an impact. So I think, you know, sadly that's going to happen more and more as on the exponential curve we're going up. The other thing, I was just at Secretary John Kerry's conference, Our Ocean, that was hosted at the State Department, and it was an international conference, and lots of, some heads of states, lots of top ministers who were there. And the focus was on overfishing, pollution, and ocean acidification. It was really exciting to see it in, you know, elevated, but the, you know, small island nations and a lot of other nations who are really dependent on marine resources are going to be more and more vocal as this gets heated up. So I think it's going to reach the popular press from angles that are other than the scientists. But it's a great question. We struggle with it all the time, and you know, the problem is so often the funding gets cut for the outreach education aspect of it, you know, because that's always the first thing off the chopping block. And in NANUS, we have worked a lot with the shellfish industry, and then they in turn go back to DC or go down to Olympia and lobby. So that's been a nice way to, we're the honest broker, we're providing the data, the information, and then they're the ones who say, hey, this is not working for us. So, yeah. Sure. But we need help, so people have ideas. Oh, Maureen and then right there.
So I had a question. In the film, they were saying that they were starting to have to treat the water for the larvae development.
Yes.
And then, do they then take those treated larvae and seed them back out into a natural environment, and then what happens?
Yeah, so Maureen's question is, in the film they talked about treating the water as they're settling the larvae, but so then her question is, once you take them out and you put them, then what happens? And so it has to do with that critical stage. So when you become, you know, something more than a couple of cells and you start putting down that first layer of aragonite in the dehinged stage, and then it's just like a tree ring. They just keep putting it over and over and over. And so as they get to a certain stage, they can better withstand the low pHs and low carbonate values. Obviously, there's a limit to that, but the larval stages are the real sensitive ones, and that kind of just fits with what we know about biology. Put a baby on a sidewalk, they're not going to do too well. So yeah, so there's that real critical larval stage that we're trying to protect. And yeah, up here.
I had a question about your data that you collect. You said you take or worldwide samples, etc. Do organizations other than the OA use that information? How do you go about sharing that information?
Yeah, so beyond OA, right? Yes, so the question is, as we're taking data globally, we're going to be taking data besides just OA variables. We take temperature, salinity, oxygen, nutrients, things like that. How about getting that to have other uses? So that's where folks like IUS, which is part of this global ocean observing system, are really able to make some big strides because, for instance, with NANUS, we're collecting OA data, but we're also collecting a whole bunch of other data and making sure that it's available for a lot of different uses. So even though someone may come to the Global OA Observing Network data portal once it's fully populated, right now it's just showing metadata. If you click on that map, you'll just see who's the source for it, but you won't actually get the data. Ultimately, you'll be in a setup like how NANUS works, where you can actually, when I showed the, let me do that right here. Right here, this little arrow, if you click on that, that says, do you want an Excel file, do you want an ASCII file, do you want whatever? And so you can actually download it onto your desktop. So the whole thing is, behind these ocean observing systems, is to get the data and make it available for many, many uses. And you know, right now we're really trying to emphasize the OA observing network because that's critical, but those data would be available to anybody for other purposes. And then the other thing is that, for instance, there's a system called IGMETS. I can't remember what it stands for, but anyways, they are doing a biological time series, so they have a lot of plankton data, but they don't have any OA data. Well, with the Global OA Observing Network, we want to be able to then put sensors at those sites to continue those time series so that then they'll continue to look at whatever they were looking at, but we also have the additional variables. So yeah, we're really working to have ocean observing systems that cover a lot of variables. Yeah, and in fact, one of the criticisms of IUS has been that, oh, you're trying to do too many things. It's like, no, we're really trying to have comprehensive observing data about the ocean because it does so many things for us. That's a good question. Yeah.
So you talked before that photosynthesizers will actually be doing better in these cooler conditions because of elevated CO2 within the water and whatnot. If you increase the amount of photosynthesizers in an area, would they be able to have a mitigating or significant mitigating impact on local conditions?
That's a really good question. So the question is, if we increase photosynthesizers in a local area, would it be able to have a mitigating factor on ocean acidification? And that is a question that I'm not the proposer, but I'm on a team who proposed that actually to the Paul Allen Family Foundation. And that proposal has been reviewed and we're waiting to hear back to actually test that in 2 or 3 different embayments in Puget Sound, have these big rafts that have algae. Then the algae are out there and then you harvest it and then make a product, whether it's fertilizer or oil or whatever from the actual algal biomass. So it's kind of that win-win. You're trying to mitigate a local situation and create a product, and then also create awareness so that people understand that, hey, this is our natural system and we're part of it, and how we interact with it can make a difference. So I don't know. If you know Paul, give him a call. Yes.
So on your shift-based series that goes out every 10 years or so.
Yeah.
Who funds that? And is there any chance that some of these global partnerships that you're making might be able to come up with funding to do it a little more frequently or add to that?
Yeah, so Maureen asked about these repeat hydrography cruises that go out once every 10 years. Two questions. One is who funds it, and then secondly, is there a chance to step that up so that we get better information, or so that we get information more frequently? And so those are internationally funded. NOAA is a partner. There's European science organizations, Asian science organizations. So they're funded internationally as a partnership. And then one of the big reasons by creating something called Go On, which is basically we're creating something out of nothing and rounding things up and calling it a system, because we really want it to be a system. We want there to be consistent methods, we want there to be consistent access, and we want there to be, able to address those three goals. One of the reasons that it's really important to do that is once you have that, then people will say, hey, we gotta make it better. And I was really excited, I have to tell you, that Go On, the very first workshop, NOAA decided to lead the effort to try to build Go On. They contracted actually with the University of Washington to hold the very first workshop. It was held down in the School of Oceanography. We came up with the name Go On. We established those goals and everything. And then at Secretary Kerry's conference, to hear him talking about, we need this Go On, So, well, I mean, it wasn't that one of the panels was, Go On was presented. So Phil Williamson from the UK presented what Go On is and why it's important. But just to hear him say that, was really exciting. And as, the people in other nations do that. And part of having a Go On is, if you're in nation X and the scientists there go to your decision makers and say, I'd really like to start an OA observing network. I think it's really important for the nation. Can we get some funds for this, that sort of thing. And they go, well, what's your plan? Well, I'm kind of thinking about this. Whereas, okay, 100 scientists from 30 nations came up with this plan, and everybody's sticking to it, and and this nation's pledged $9 million, and this nation's doing this. What are we going to do? It changes it. So I really hope so. I really hope so. And it's been so hard, with the economy, the budgets and everything, so a lot of the sustaining measurements have just got curtailed, but we're hopeful. Cool. Yes?
I just have one other question. As you're funded by private industry at times for some of the projects, does that affect how effectively you share data with other scientists? Does that impede any communication due to wanting that proprietary knowledge or something like that? How does that work?
Yeah. So the question is, if we're funded by private foundations, is that going to impact the data availability? And the answer is no, because we're not going to take those kinds of dollars. And so in fact, Go On, we've created something called Friends of Go On, and there are 5 foundations that it was announced that are going to pledge support. We don't know how much, but they're interested. But no, that's one. If you read the plan, which is on the Go On website, that's one of the basic things, is that the data will be shared. And usually foundations want you to share it. It's the scientists who are reluctant. It's like, I don't publish on it. And we talked about impediments to data sharing, and so and really trying to get from an older time when I was scientist X and I just published my papers and I got notoriety for that, to a different world where we're working collaboratively, that my data supports this, and now we're writing these synthesis papers that are pulling out more knowledge. And so there's that. I think one of the bigger problems will be national barriers to releasing coastal data from some nations, and we heard that from some of the scientists participating. But yeah, we're just not going to take any foundation money that would ask us to do that.
Good time for maybe one final question.
Yes.
As the enterprise support equipment develops, as we have better automation, is there any consideration to using some of the other ships that are all over the place to collect data and transmit it? I mean, there are commercial ships, there are cruise ships, there are heaven knows how many. In an age of citizen science, it seems like.
Yes. Yes, so the question is a really good one about, as we get better technology, better sensors, more economical sensors, are we going to utilize other ships, whether they're commercial or luxury or citizen science? And the answer is yes, yes, yes. And so, as you see, we already have this volunteer observing ship system. There's an entity called Seakeepers who outfits luxury yachts with sensors. And then, yes, there's been a lot of people saying, well, citizen science is really important, and it's just really hard. Sort of, well, okay, you got 20K in your back pocket, because the measurements are difficult. And so as we get better sensors, that will make a big difference. But there is a role for citizen science in the biological observations, right? Whether that's plankton or seagrasses or, just really trying to connect with getting better observations on the biological responses. And that's not expensive, it's time intensive. And that's where we really need some better systems. And I think automated databases can help with that. That's one of the things Dan Hughes would like to do, is a database. Like right now, our database it's one way. You can get the data that we're providing, but you can't put yours on there. And we're hoping to get that funded so that we could go forward with that. Because I really think that, even with this starfish thing, which is not related to OA, I hope you heard me say that, at least not that we know about, because everybody, oh yeah, I heard Newton saying that. I was, no, I didn't. They are asking for boaters to help with the observations on where are the starfish, where are they finding them, where are they seeing the ones that are decomposing. So that's really important, that citizen science role, and let's hope we can push forward on that. Thank you. All right. Thank you.
Our coffee and snacks have arrived, so please feel free to enjoy the break, and we're back in here at 11 o'clock for our next speaker.
Yeah.